So today I'm going to talk to you a little bit about exploratory data analysis. I've been doing data analysis in genomics for over 10 years, and to me this is something very, very important, and so many times it saved me wasted effort on building models when you find out that the actual main trend in the data is some batch or some technical effect that you actually need to remove before you start. So hopefully I'll convince you that there are multiple ways to do exploratory data analysis, so there will probably won't be one that you will like best, but it's always a good idea to throw a couple of different approaches. With exploratory data analysis, there's no right and wrong answer. You're literally just trying to get a feel for the data, and hopefully I'll bring that forward.
Before Aedin goes on, just so you know, Aedin's put all the slides and stuff on the website, so don't have to scribble notes.
Yes, they're on the resources folder. There's a folder called My Name under resources, and on that I've put all the slides up. So this is, where do you start these days when you actually want to introduce something? You start with a definition, and where produces definitions? But Wikipedia. So this is the Wikipedia definition of exploratory data analysis, an approach to analyzing data sets to summarize their main characteristics, often with visual methods. I actually quite like Tukey, who actually wrote a book in 1977 and really brought forth the idea that you should do exploratory data analysis prior to doing predictive modeling. But his take on exploratory data analysis was making pictures such that you could look at them with the picture examining eye. And this is a picture here of Tukey. The greatest value of a picture is when it forces us to notice something we never expected to see. Fortunately, that's normally a batch effect, but sometimes you see interesting biology that you mightn't have expected. Sometimes the main, I was looking at co-culture data recently. We're looking at 3D co-cultures of breast cancer cells and accessory cells. And the clinician that I was working with was really expecting to see that the culture substrate might be the most important thing, or maybe it was the estrogen that we were targeting with, or was it the actual different accessory cells? And exploratory data analysis really gave us the idea of, well, which is the most important, pulling out what were the variant trends in the data. And this is just his book, which I'm actually, from the 1977, is still in print and has a very good Amazon review. You can buy it for $80 on Amazon. So it's actually pretty impressive that a book from 1977 is still in press. So why do we need exploratory data analysis in genomics? I think it's pretty clear, but we are dealing with multivariate data. And really, it's very difficult to just eyeball this, using Excel. We really do need to use more sophisticated methods. If you can't read this, that says, I think I found the corner piece. And this is the 3 billion piece human genome puzzle. So as I mentioned, we've got genome scale data. The data is multivariate. We're dealing with tens of hundreds of thousands of variables. And so exploratory data analysis will allow us to get a handle on the data. Exploratory data analysis should happen as first part of a larger process. And it should take place before the more rigorous statistical analysis. Why is this? It's because it's really our first line of defense against bad data. Okay, we really want to do the exploratory data analysis, pick up our batch effect, correct it, and move on to our predictive model, and not waste time on doing a wonderful training and test cross-validation of thousands of variables when really it means rubbish. Bad data in, bad data out. But hopefully it may lead to new insights, new questions, or feed into the process of predictive modeling. It can help us decide what questions to ask and detect confounding covariates. Oh, sorry, I'm going the wrong way. This was an interesting, I don't know if you read different bioinformatic blogs. This is Jeff Leake's blog. Do you read this, Sean? I do. And this was quite an interesting discussion that went recently, and it was actually picked up on Twitter feed. You're much better on Twitter. I started it and then particularly stopped. But this lady, Hilary Mason, actually put forward this question of what you do when you get your hands on a data set. and it was quite an active Twitter feed. And Jeff Leake actually made a really nice summarization of what to do with the data set, given it, here's your data, what do you do with it? So Jeff's first step 0 is try to figure out what you're doing with the data. So stop, look, think. Ideally check in with the person that generated the data. What do they really want from it? What do they expect? How was the data generated? Do they have any concerns about how the data was generated? Was the data generated over multiple years, multiple dates, multiple different batches of RNA extraction kits? Maybe find out if there's something in advance that they mightn't have thought to tell you, but that may actually be a source of variance. Learn about the elephant. This is just feel around and see what I have. Bring in the data, do head, tail, feel, find out what are the column names, just to get a feel of what you're talking about. Next thing, Jeff suggests clean and organizing the data. This is something I definitely spend a lot of time doing. Fixing NAs, imputing the data if necessary, or removing columns with a lot of NAs, and as Jeff said, swear a lot. You try not to, sometimes it's inevitable. And the next thing was plot that stuff. And this really is the exploratory data analysis. So for simple univariate data, or where you have only a few variables, or even when you have multiple variables, sometimes using histograms, scatter plots, density plots. I'm sure you've done quite a few of these already. Some? GSHL 1-2 plots, different things like this. Have a look at the variables one by one. And for multivariate data, do some sort of dimension reduction, principal components, or some sort of cluster analysis. And after that, you can get your first quick and dirty answer to the hypothesis that's being addressed by the data. Do some simple predictive modeling, see if you're in the right ballpark, and go back to step 0 to find out if it's actually in the right direction. So for univariate structures, box plots, scatter plots, MA plots, where you plot either the difference or the variance versus the mean to see if there's what's the structure between the variance and the mean, if they're dependent. Pairs plots, correlation plots, these are all very, very useful things to do. I think Rafa may talk about these a bit more anyway as well. What I'm going to talk mostly today about is actually clustering and ordination or dimension reduction analysis. I think you've already had some introduction to clustering, so I'll breeze through those slides really, really quickly, or skip them all together. Which would you prefer? Breeze through? You want to recap? Okay, cool. And then I'll go through dimension reduction analysis and ordination. Okay, I'm sure you're familiar with the de facto genomics plot, which seemed to occur in nearly every single paper for so long. These have since been sort of taken over now by spaghetti diagrams or network graphs. But I'll go through later on, particularly in the tutorial, some of the different packages for generating these, and I'll concentrate a lot as well on different packages for doing ordination. So clusters can be built bottom up or top down, agglomerative or divisive. Agglomerative, you start with one, you add in another, you add in another, you add in another. Okay? So you slowly build up your network of friends, or your clusters. Divisive, put everybody in the same room. So you start with everybody, and you start chopping down into groups. K-means is a typical divisive group. You put everything into, into one big cluster or into two big clusters, and you start chopping down. Okay, for agglomerative clustering, hierarchical clustering is widely used. Whatever clustering method you use, what you will see is dependent on the distance measure used. So you will see what you seek to explore. Okay, so the choice of metric is fundamental. It's really know what you're doing. So rather than publishing a paper saying we did hierarchical cluster analysis, say we did hierarchical cluster analysis with a 1 minus Pearson correlation coefficient distance metric, which was joined by average linkage. Okay, specify what you did, and then somebody can repeat that. You're probably going to cover reproducible analysis and Yeah. So we did a study, it was published in Nature Genetics a couple of years back, on how many studies could be reproduced. And it wasn't as good as what you might have expected. There was a lot of studies where we could get the data, but we couldn't reproduce it because the methods weren't fully described. And so it's really important, and you're in a fantastic opportunity here to learn how to script in R and then actually provide the methods and the code directly to everybody that's reading your paper such that they can reproduce your paper. And after the scandals that happened at Duke, that I'm not sure you're all familiar with, and
We haven't talked about it.
You haven't talked about it?
Sure.
I have a great, do you know what I'll do is I'll bring up the slide later. I've got that great slide from the day in PubMed when all the papers were retracted. I actually did a screenshot of it. It was, whether it was, you know, if you listen to Keith Bagley, they say it was kind of premeditated, you know, falsifying of data. These things can happen. You know, when people particularly are very, very keen on getting a paper out and with the pressures of today. But there was some systematic things that happened in the analysis of NCI 60 data whereby they said that they developed classifiers to predict chemosensitivity to drugs. And when Keith and others, and actually I did it as well at the time, started analysing the data, we were going, something's wrong here, it doesn't mishmatch. But Keith actually followed it up systematically and said, right, actually, let's get the data, let's reproduce what they've done. And it started off by just being point errors where all the data were out by one, so all the probe IDs were incorrect. And then simple things like where they were actually doing sensitivity to leukemia. Leukemia is generally pretty chemosensitive. They actually were doing their ones and zeros back to front, and they were saying it was mostly chemoresistant. So there was a lot of errors in their analysis, but from that they actually developed a set of biomarkers and they went on towards clinical trials. And the clinical trials were starting in multiple different cancers based on these papers that were fraudulent, they were wrong. And it's led to a big push to sort of say, well, you know what, if you actually want to pursue these biomarkers, make sure that your analysis is completely and utterly transparent. And that's very much where we work with, we try to provide scripts and so show exactly what we've done. But I have that, where all those retracted, like really high profile papers. It's very, very sad. So I went on a tangent, I'm sorry. The choice of the metric is fundamental. Describe what you did. Okay, here's a pop quiz. Which do you think is closest? We've got like 8 genes or 8 proteins here. What do you think is closest?
Exactly right. I did the same thing yesterday. So.
Oh, did you? Is it the same?
Not the same picture.
Different colours. It's completely right. If you maybe want to look in the closest distance, the Euclidean ruler distance, you might think 7 and 8 are really close because they have the same. Or if you're looking at maybe a Pearson correlation and you're looking at trends, you may think, well, actually 1 and 2 look interesting. It's up to you. It depends on what you want from the data. Okay? So the distance will be dependent on what you see will be dependent on what your distance is. So the Euclidean distance here for these is very different to their Pearson correlation. Okay. Euclidean distance, probably remember your Pythagoras theorem from, that's a case of the Euclidean distance. It's the straightforward ruler distance. This is not Euclidean distance. If you ever got a pub trivia, can anybody say what was wrong there? It's not an isosceles, it's a right angle triangle. Yeah. Does you know when the wizard gives him his brain and he goes, the sum of the squares of any 2 sides, and he thinks I have a brain, but he doesn't. Pearson correlation, you're literally trying to do a line through your points. If they're completely correlated, it'll be 1 or -1. If there's no correlation, it'll be 0. Be careful of correlation coefficients. Okay. Sometimes if you have got one point, particularly if it's like larger numbers, it can completely throw a correlation coefficient. So when you actually get on then to multivariate methods, the rely on correlations or doing canonical correlations analysis, if any of you heard of it, those can be susceptible to overfitting as well because they actually maximize correlations. Okay. Different distance metrics, lots of different properties on each. I'm sure you probably spoke. Okay, I'm not going to go through this with you. If you do, the slides are there. I actually do a back of the envelope. It's the simplest thing in the world to do, is to calculate a hierarchical tree using a calculator. It's very, very simple to do. And don't be put off by just the fact that you're pressing a button and it's doing it for you. It's very, very simple to do. So if we were to calculate the Euclidean distance between gene 1 and gene 2 here, what's the distance? Can anybody tell me? What's the difference between gene 1 and gene 2 in experiment 1? What's the difference in experiment 2? What's the difference in experiment 3? Experiment 4, 5, and 6? Okay. Each case, square it. So 1 squared, 1 squared 1. Sum it up. 5. So the distance is the square root of 5. Okay, so it's minus 3, minus 2. The difference between them will be 1 squared. Okay, 1 squared, 1, 1, 0, 1. Add them up, you've got 5. It's the square root of 5. Okay, which is 2.24. What's the Euclidean distance between these two genes? You can say the square root of what number? 3. See, you've calculated, it's easy. The square root of 3. Then essentially all you do, if you're doing single linkage, you just take the minimum. So this is our distance matrix between all of those genes. So you should see here between gene 1 and gene 2. Have we got a pointer? No. Oh, sorry. Gene 1 and gene 2 is the square root of 5, 2.24. Gene 1 and gene 3 here is the square root of 3, which is 1.73. So calculated the whole way along. Then to join the tree, we literally just take the minimum here if we're doing single linkage. So what's the minimum of this? I have it highlighted. It's a hint. 1. So we now just join those 2 points together. Okay? So now we join 5 and 6. Okay? We join 5 and 6 together. We now take the minimum of each of 5 and 6. We have a new row here. And in this case here, I've just joined. Sorry, I missed one there, didn't I? We take the minimum now of 5 and 6. So the minimum here is 0. The minimum here is 6.78. The minimum here will be 6.78. Okay, so you'll see that here, 6.78, 6.78. And we continue basically to take the minimum. And then, in this case here, it's 1.4, so we're going to join gene 4 and gene 5 to 6. And here again, we just decompose this down. What's the minimum here? 9 or 0, 8 or 1, 6 or 7. And you continue doing this, basically making it smaller and smaller. Yeah?
So actually, you do the join 5 and 6. I'm not sure I follow, like, what's the math that you're doing there for the joining of 5 and 6? Or you just taking the minimum?
You just take the minimum.
Okay.
That's it.
So 6.78 for their relation with 7 and 8.
Yep. So for single linkage, you just take the minimum. That's all it is. You just take the minimum.
Okay.
And so what's the minimum between, if you're joining you're merging here 4 and 5 and 6. So you're adding this node on here. What you simply do is, well, what's the minimum here? 0. What's the minimum here? 6.78. What's the minimum here? 6.7.
How do you calculate the distance from 4 to the joint 5 and 6?
It's just whatever that distance was. Let me show you the tree. Be easier to see. So can you remember what was the first nodes we joined and what was the distance between them? 5 and 6. And so if you read off the tree, what's the number here? What was the next one we joined? We joined 4 to, and what was the distance? It was 1.41. Let me go back so you can see. It was 1.41. 1.41. And so you read it off the tree. Yeah? There's the slides there. I recommend just take the piece of paper out and just do this. Literally, you can do it in the back of an envelope.
So if you're clustering samples,
Mm-hmm.
you have to sum up every single gene, and then take the square root of every single. How does that?
So if you want to get the Euclidean distance between two genes, you would take gene 1 and gene 2, take the difference, get the square. Next gene, gene 1 to gene 2, gene 1 to gene 3, gene 1 to gene 4, gene 1 to gene 5, gene 1 to gene 6. So you get a triangular matrix. Okay, so you get the difference between every pair of genes. So that's actually a very important point when you're doing our clustering. You're looking at the pairwise distance. So you're looking at every single pair. What's the distance between that pair? One thing that was important to notice there, and I'm not too sure if I brought it forth, if you look here, these actually are at the same level. And that's because when we got to here, after joining 1, there was actually 2 cases of 1.41. So the distance between gene 2 to gene 3 and gene 4 to gene 5 was actually 1.41. Which you do first is actually pretty arbitrary. Okay. So, according to this, in which case I've done Euclidean distance and single linkage, this is our tree. And all the calculations are there for you to do and repeat yourself. We can see that we actually have. Now, this is the second thing. When people look at trees, how do you start looking at them? Do you look down here or up here? Okay, votes for up and down. Who tends to look up here and who tends to look down here? Sorry, I should. Who looks up at the top? Who looks down at the bottom? You got to learn some stuff. This distance here, this ordering here is, in most clustering methods, is completely and utterly arbitrary. Okay, I like to think of a cluster diagram as a kid's mobile. You know, I think it's in the next picture.
If it's rooted or not. Because I mean, if it's an unrooted dendrogram, then it matters less what's at the top. But if it's actually rooted, then it matters more what's at the top.
Yep, but even the ordering here, whether it's 3 and 6 or 6 and 3, is completely arbitrary here. So sometimes people read these and say, "Oh, 6 is beside 7, and 5 is beside 8 here." You can flip this completely and flip this in itself and flip this in itself. These can all be, so although what you're looking at is the structure from the top, so think of where the tree is hanging from. These little things here can be flipped around happily, and you can do it to visual. A lot of people will do it so that it looks like a prettier tree. But this tree is the exact same as that tree, even though 3 and 6 are now flipped, and these 2 are here, flipped here as well. Okay, so when you're looking at a tree, start looking at the top and see what the structure is. Okay, the next thing to do is say, well, actually, how many clusters does it look like I have? Okay, and in this case here, you can say that you have 2 clusters, or perhaps you say you have 3 clusters. Which is right? Well, there's lots of different approaches that you can use to look at like cluster compactness, or you could do bootstrapping, where you try and measure the robustness. But perhaps for initial exploratory data analysis, it's just one hypothesis. There may be 2, there may be 3. Let's do some other methods, see what else we see. Okay. So I actually skipped a slide there, which was we joined using the minimum distance, and that's called single linkage. You can also join by the maximum. So you start with the maximum distance, and you join those nodes. And this is the maximum. Or you could join by the average. You will see that you get quite different trees depending on what linkage approach you use. Centroid or average linkage is probably the most widely used. With single linkage, you tend to get this chaining. With complete linkage, you tend to get like over large clusters. We mentioned already that when you're looking at hierarchical, sorry, what's the question?
In the previous slide, the one that you just showed, you have different trees, but it seems that the heat maps are exactly the same.
It's the same data. Oh.
So you're saying that if you're using minimum, average, or maximum, trees are different, but the heat maps are slightly.
There's slight differences. There is slight differences. This data is very it's a data. This is actually John's slide, John Quackenbush's slide. But yeah, it's very much fake data. You know, all down, up, down, up. Nothing happening. So but there is, if you notice here, it's probably more. This one here is this one here, and that one there is that one there, and that one there is that one there. There is slight differences between them. But this is like a really easy case of clustering as well. So hierarchical clustering forces samples to forces a hierarchy on the data. If you throw data at hierarchical clustering, it will form clusters. Okay, so the fact that you get 2 clusters or 3 clusters, that's its job. It will find clusters. Okay, so it will compare samples in a pairwise manner, and sometimes it can be difficult to visualize if you've got a lot of data. Sometimes forcing things into bins isn't maybe what you want to do, particularly if you've got maybe a dose response or some sort of gradient, and you really know that there's probably more of an overlap rather than distinguishable clusters. And a complementary approach to that is actually using ordination. Now, unfortunately for ordination, dimension reduction, latent variable analysis, eigenvector analysis, factor analysis, there's multiple ways by which the same thing or the same approach is called. And it depends on what branch of computer modeling or statistical analysis is your background, or if you're from France, because they've got their own terms altogether. So there's lots and lots of different terms, but essentially you're trying to visualize data on a fewer number of axes. So find the axes onto the data onto which the data can be projected, such to explain most of the variance in the data. This is not what we're talking about. In the genomics literature, this is the kind of pictures you tend to see, where you tend to see this plot of one axis versus the other. In general, you tend to put the more variant axis, so the axis that represents a bigger trend in the data, on the horizontal, and then the next variant trend on the vertical axis. And you're simply looking at an X by Y plot. These are very complementary approaches, because with cluster analysis, you're looking at the pairwise distances and similarities between objects. In ordination, you're looking at the variance of the whole data set to highlight trends and gradients and patterns through the data. Okay, and some publications present both. What are you doing with ordination? Well, giving a cloud of points, for example, in this case here, if we just have very little baby data set of 3 genes, not that you're ever going to really get that these days, but if we had like our 3 favorite genes and we joined we created an XYZ plot of them, you're trying to find a linear line such that it captures a major trend in the data. Okay. What does that look like if you're thinking in terms of gene expression? More than likely you're trying to find where there's a lot of genes doing the same thing. Okay. That will be a big trend in the data. So if you've got 500 genes going like this, and you've got 200 genes going like that, and you've got 100 genes going do, whatever, that probably would be your PC 1, 2, and 3. Okay, so it's a correlated trend in the data. Okay, so the first axis will capture most of the variance or most of the information. The next axis is projected such that it's at a right angle, it's orthogonal to it, or decorrelated. And you continue through the data, finding another axis that's uncorrelated and represents another trend in the data. The number, what do you think is the number of vectors that you could get from this data set here. Yep, the normally number of genes minus 1, or the number of samples minus 1, whichever is smaller. So if you have a 30 by 300 data set, you should be able to represent the trends on 29 vectors. Okay, so I think I said that already. So each axis represents a different trend in the data. And when you're looking at these, in this case here, I'm looking at 3 different species. We're going to analyze this data set later. This data set I've been analyzing for years as a tutorial, but I think the idea of species is very easy concept to grasp. So the first axis here is, what is it doing? What's the trend that it's representing? Yes, species, that humans are different from bonobos and gorillas. Are humans more different than bonobos or gorillas? Which one is further? Gorillas are further apart. That's exactly it. You're looking at the furthest points from the origin when you're looking at these plots. So you're looking at the trend of what samples are being distinguished, and the samples that are driving that are those that are furthest from the origin. Anything projected in the same distance from the origin tends to have the same genes that are up and down in it. Okay, so you're projecting axes through the data which capture the variance. Each represents a different trend in the data. They're orthogonal or decorrelated. They're ranked, so the first axis is most important. And these axes tend to go by lots and lots of different names, okay, depending on which nomenclature you use. So principal axes, principal component analysis, latent variable analysis, factor analysis, eigenvector analysis. So given a data set, you can see the amount of information that's represented on each axis. In this case here, this here is telling us, I can't actually read my axes here. I think this is maybe 10%. So in this case here, we've more than 10% represented on the first axis, and the next we've got, I don't know, 7% here. So this is the amount of information that's represented here. And from these analyses, you'll get 2 projections. You'll get the projection of your samples and the projection of your variables. So your arrays and your genes in this case here. These are projected in, obviously, in the same space here. So if you want to find out which genes are expressed in humans such that they're driving this pattern, you will look in the same direction from the origin. These will be the genes that will be highly expressed in human. If you want to find the genes that are highly expressed in gorilla, you go to the gorilla end of the axis. The genes here will be highly expressed in gorilla, and these genes here won't be expressed in gorilla. Okay, so most of the eigenanalysis are actually computed using singular value decomposition. Okay, this is just the eigenanalysis that's used. Given an initial data set here, you decompose it to 3 matrices. One which contains the sample coordinates, or the cases, one which contains the features, and then a diagonal matrix which just tells you how much variance is in each vector. So this is the first eigenvector here associated with our samples. This here is the variance associated with it, and here, this is just transposed, is the trend that you're seeing in the genes. Okay. How to choose how many eigenvectors you should look at? This is a question like I asked myself, and lots of people ask, is, you know, I've done my PCA analysis, I've got my plot here of my scree plot. How should I look at axis 1, 2, 3, 4, 5? Where should I stop? One cutoff that people use is to look at 70% of the variance and see how many genes or how much axes are required to capture 70% of the variance. In this case here, since PC1 represents 40%, it's going to be done pretty easily, at least to see 40% of the variance. And this is 25%, this is 20%. You could just look at 70%. Another approach people use, these plots are called scree plots. And scree plots is that moraine that's kind of at the bottom of the hill. Well, you can kind of take a cliff jumping approach which is if you're at the bottom of the top of the cliff and you jump, where do you land? And so here you would land straight down here, and here you would probably land around here. That's probably a good rough guesstimate of how many you should use. Again, this is exploratory data analysis. It's up to you to explore. It's up to you to work out exactly what you want to do. Ordination methods. The most common is principal component analysis. I tend to like, correspondence analysis a lot. I think Sean has done multiple dimensional scaling with you. A principal coordinate analysis is decomposition of a distance matrix. There's many, many different ones. The association between these is how you transform the matrix in the first place. In one way, this is akin to what distance metric you use in order to do your cluster analysis. Okay? So how you transform the data initially before you decompose it will give you a different result. Okay? PCA, correspondence analysis, most of these methods can be computed using singular value decomposition. So these are often referred to as decomposition analysis. SVD was first described, at least in the genomics field, to microarray by Alter et al. I'm giving these citations at the end of the slides. Wall et al. Has a very good description of what the difference between these is. So principal component analysis is probably the most popular of the different ordination methods. It's an eigenanalysis of the covariance matrix. Many people date it back to a beautiful paper written in 1901 by Carl Pearson, which I have on my website if you would like to read it, where he really describes of how to fit lines through points in space. Others will date it to the 1970s to the machine learning field. You apply to quantitative data, first applied to microarray data in 2000. These are some of the many, many packages in R. Precomp is widely used, PrinComp, AD4. We'll have a look at some of these later. Yes, sorry.
Is there like a little definition of what
Eigenvectors are principal components. It's just that this is an eigenanalysis. You're doing an analysis on the matrix. Okay? This here is an eigenvector. This here is an eigenvector. It's just a vector, or just numbers in the matrix after the decomposition. So when you do this matrix decomposition, you end up with one, which is just the diagonal matrix. So you just don't end up with one vector here, which represents the actual variance. And then you have two other matrices. One which here is of the samples, and one which, this back to front. This here is the samples, and this here is the genes. Okay? And so your first principal component, or your first eigenvector, is just the first vector in that matrix. And you will have coordinates on that in gene space and in sample space. And you can call that your eigenvector, you can call it your principal component, you can call it your principal axis, you can call it your latent variable. It's actually the same thing, it's just the first column here.
And you're always just looking for linear relationships, right?
These are always looking for linear relationships, yes.
So the eigenvalues correspond to the variance contributed by an eigenvector.
Yes.
But is it actually looking at the eigenvalue that is the variance value, or these, like if I'm looking at an eigenvalue,
Mm-hmm.
and I compare it to all the other eigenvalues, is its proportional? Is its proportion of all those values the proportion of the variance?
Yes, it's a proportion of the variance. So in principal component analysis, this is sum to 1. In correspondence analysis, decomposition of the chi-squared matrix, it should sum to the chi-square of the matrix.
And the squiggly plot is just a plot of the eigenvalues?
Yeah, it's just simply a plot of the eigenvalues. So it's just showing you how much variance is captured by each eigenvector. I think when we do this and you have the matrices in front of you, it makes it a lot easier. What are we doing for time? How much detail should I go through with this?
If you have your script and you have your tutorial, so we just want to try to finish up with that by around 5.
Okay.
So between now and then, you decide.
Okay. So I have a lot of detail on these. I can go through it very quickly, or I can skip it. Quicker is better than skipping. You're talking to an Irish person asking her to talk quickly. I can talk quickly. Okay, so this is just showing you what the transformation is here in your column in PCA. Your column mean centering of the covariance matrix. So if you have a point in your matrix or a value in your matrix here, this here is your point Nij, where you've got J samples and I genes. The sum of the genes, just the sum of this row, is Ni here, and the sum of this column is just Nj, and this here is the sum of the total matrix. What you're simply doing, your point Xij, after the transformation here, is just your column centered. So you're just, what is my thing here? You're taking away you're mean centering it here. You're just dividing by the total. Okay? You're just scaling in one direction. You're just dividing by this here. You take this by this. Okay. PCA of NCI 60 cells, this is like the first description, or to my knowledge, of PCA in genomics field. And they performed a principal component analysis of the NCI 60 data. These are figures from that paper. They found that they looked at 5 eigenvectors. This here is the eigenvalues associated with those eigenvectors, or those principal components. And they decided to cut it here. So kind of like jumping off the top of a hill. This is kind of the bottom of the hill. And then they say these here are their eigenvalues, 1, 2, 3, 4, 5. This is the eigenvalue and the percentage of variance. So the first component captured 15% of the variance, the next captured almost 10, the next 6, 4, and 3. And if you look at the cumulative variance, adding these up, so the first 2 components would have captured 15 plus 9, so 25% of the variance. Okay. This is the NCI 60 data. These represent multiple different tissue types. There's 11 different tissue types in this. So there's a lot of different information in this, so it's not surprising that it takes more components to capture that information.
Sorry. They say 5 PCs contain most of the variance, but they're only saying 40%.
Yeah, they're only getting 40% on this, so it's not that much. So if they want to get 70% in this case, you're probably going out to 10 PCs.
But then only the first 2 ever make it into the paper.
Sometimes first 3.
Oh, right.
Yeah. So you're just looking at but then if you're looking at cluster analysis, it's only you cut the tree and you get the first 2 clusters or the first 3 clusters. It's the same idea.
Sometimes it's the second 2.
The first one sometimes can represent noise. Yay.
How do you know that represents what is?
It's not associated with anything that can be biologically defined. Or you find that it's associated with the date the analysis was done, or the assays were done. Not normally the analysis. So here they're looking at the first component, the second component, the second and the third, and the first and the third. And they show here, in this case here, that the melanoma cells here are being separated here on the second axis. And the first axis, they don't really describe it so much here, but it actually is. I've done the analysis on this. You're kind of splitting by EMT. So you're splitting by kind of the more epithelial cells from the more mesenchymal ones. On the third component here, they are splitting the leukemia cells from the carcinomas, and they're saying here on the first component, they're splitting all of these here, which they're saying are the CNS, the renal, and several others, and they kind of don't describe that one as well.
Do you infer that one component corresponds to a biological function based on you noticing how the data is behaving
You can test these as well. So you can either split at the origin, or you can look at the linear line and say, is that now associated with any of my biological variables that I know about? And that can also be used, so surrogate variable analysis that Rafa will describe probably next week is a PCA approach to actually looking for batch variables and removing them. So considerations when applying PCA, you're decomposing a column-centered matrix, and the distance you're looking at is more akin to Euclidean, is a Euclidean distance. Okay. It's robust, but designed for analysis of multinormal distributed data. If your data is very skewed, or if there's a really big scale effect on the data, that will be evident on the first component. And you may well be going down to 3, 4, and 5, and 6. I know a colleague of ours, when they've been analysing some paraffin arrays, so microarrays done from paraffin embedded tissue, quite a big study that was done on colon cancer from the Nurses' Health Study. They actually had to go down to, I think the 4th axis was the first one that they were getting something biological. 1, 2, and 3 were all noise. So don't assume that the first axis is always biologically informative. If you've got a big scale effect, this isn't important for log 2. Sometimes you may have to row center the data. I tend to like correspondence analysis, and I'm going to describe it why. It tends to handle zeros better, and you're dual scaling the data, so you don't get the horseshoe effect that you sometimes get with PCA. It's my cartoon to break the session. If I'd known they wanted me to use all this information, I'd never have asked for it. So correspondence analysis is a lesser known form of dimension reduction. It's an eigenanalysis of the chi-square distance. So because of that, it measures the strength of association between a gene and a sample, or between a feature and a variable, feature and a case. Again, this is an approach that's been described multiple times and has multiple names, such as reciprocal averaging or ordering or dual scaling. It was originally designed for analysis of count data, so it assumes that the data are positive and that they're counts.
What does that mean?
If you have got negative values there, you have to scale them up such that so that you've no negative data. So you just normally transform, like add the minimum, so get the minimum number, we'd say -3, so just add 3 to the thing to scale it so they're all positive, is what I tend to do. What you're doing here, this here is the, if you have a sample Nij again in our matrix of I genes and J samples, what you're doing here is you're scaling Nij to be Xij, and Xij is you take away from the expected value. So this is your observed value, sorry here. This is our column weight, this is our row weight, and this is the weight of the sample of its contribution to the matrix. In this case here, this here is the column weight here is basically the sum of that column divided by the total of the matrix. The row weight here is the sum of the row divided by the total, and our point here is basically that point divided by the total of the matrix. So this is basically the contribution of that sample to the matrix, the contribution of that column to the matrix, the contribution of that row. Your expected value here is the product of the row and the column weight. This is your observed, it's your observed minus the expected to get your chi-squared transformation. So you're scaling this point both by what you expect to observe on the row and what you expect to observe on the column. Okay. And essentially it measures the strength of association then between the gene and the sample. Okay. This dual scaling is very effective, I find, when you've got noisy data, because you remove a lot of the strange sometimes scalings that you see, particularly if you've got proteomics data sometimes where, you know, half the data is albumin. You do need to scale in both directions, so dual scaling of the data makes sense. Okay, whereas just dual scaling makes a lot of sense to me, and intuitively this makes sense to me. First described by Kurt Fellenberg in the literature in 2001, and they looked at gene expression of yeast in different cell cycles. And the nice thing about this is you can plot the genes and the samples in the same space here, and every sort of nicely colored square here is an array, and the dots are the genes. This here is the origin. So this here is 0 here on axis 2. This here is the first component, second component. This here is your origin. So taking out from the origin here, you actually observe the different phases in the yeast cell cycle. And here at the stationary phase, you're seeing a lot of histone packaging genes that are up. So you can plot the genes and the samples in the same space. Genes and samples, those with the greatest weights in that space are those that are projected furthest from the origin. A gene and a sample which are projected in the same direction from the origin indicate a gene or feature that has an increased value in that sample. So an upregulated gene in that sample. Okay, and then they pulled out each of the different axes and showed that these were associated with different stages in the cell cycle. Okay, when applying correspondence analysis, it originally was designed for count data, so the same units. It has to be non-negatives, or you may need to make a translation of the data. It does really well with lots of zeros, so I tend to like it for count data such as RNA-seq. It can handle quite sparse data where there is zeros. If you have PCA and you give it a data matrix where there's a lot of zeros, you get this crazy kind of arching kind of effect with the data, which isn't so useful. You're dual scaling the data, and you tend to get some of this noise that you tend to get in PCA, particularly some of the first axis. You don't tend to see it so much because you are dual scaling. Other methods, independent components analysis is a really nice approach. It doesn't force orthogonality. So the axes don't have to be decorrelated. So sometimes you can get more subtle trends. But because of that, the first axis isn't necessarily representing the most variance. I've got a plot of that which will describe in a second. Spectral map analysis, there's a whole load of these spectral type names of different types of analysis, and these tend to be related to correspondence analysis. And I've some different citations in the back of this talk which describes these. Multiple dimensional scaling is scaling of the distance matrix. Classical MDS is also akin to principal coordinate analysis. You make your distance matrix. For example, the Euclidean distance matrix we made earlier, where we looked at the difference between each of our two genes, and you got the Euclidean distance, you've got that triangular matrix, and then you decompose it. That's principal coordinates analysis, and that's related to dimension scaling. I mentioned independent component analysis here. You're not constraining the axes such they're orthogonal, but what you get with this is you have to then try and work out which axis is what you want. These colors are horrific, I apologize. This here is our first axis, our second, our third, our fourth, our fifth, up to the 10th. It's going up to back. This is axis 1 and 2, axis 1 and 3, axis 1 and 6. You kind of have to look at the axes to try and work out which pattern you're interested in. It's not necessarily ranked. So sometimes that can make it more difficult to analyze, but it can pick out more subtle trends in the data. After looking for the actual clusters that I actually want, you can see here, this here is looking at that same data set with the humans, the bonobos and gorillas. This is the result from FAST-ICA. In this case, I've picked axis 6 and 9. This is my correspondence analysis, picking axis 1 and 3. Okay. It's kind of like apples and apples. Yeah.
I'm probably going to get to this, but assuming your first principal component explains a ton of the variance and is something you're totally uninterested in, is it relatively straightforward to then remove that effect before you do the rest of your analysis?
Yes, you can remove it. That's exactly what surrogate variable analysis does. Yeah, you can remove it. And very early in the literature in sort of microarray analysis. Who was it that first proposed it? Was it Tip Sharani? Or is it Steve? Was it Alter? It was actually proposed very, very early on as a method for actually normalizing microarrays and removing noise. But because you may be removing signal as well, it was never actually taken on. But yes, you can remove that if you want. Or you can just say, do you know what, I'm just going to ignore it and I'm just going to start from axis 2 and 3 and look at axis 2 and 3. This is exploratory analysis, so it should tell you something about the data. There's no hard and fast rules of what's right. So just to summarize here, singular value decomposition is the eigenanalysis, the way in which you actually take that one matrix and break it down to those other matrices. Okay? It's a way of computing principal component analysis, correspondence analysis, and many of these other dimension reduction methods. And there are multiple different packages within R for doing these, and I will describe some of these. I have all the literature on this here. And hopefully, the important thing is that you test your intuition about the data. Okay? Keep an open mind. Expect to find the unexpected. You'd be surprised what you find. And sometimes you do have to go digging as to what that means. And sometimes, you know, the person who did the analysis doesn't know the date that they were done. But sometimes if you read the header files in the actual data that you have, it has a date hidden in there, and you can extract that out. Sometimes that's what you find. Be curious, be skeptical, and have fun. Okay, I have a section on... okay, sorry, this was... Exploratory data analysis should happen before predictive modeling. Look for the obvious. To try and avoid the errors that you might find in your predictive models. Okay. Expect the unexpected. This is me like going through Google Images and picking out things that I thought that were fun. I have a couple of case studies here, and will I finish with that? And then take a break? Okay. So this is just things from my own analysis that we found. Okay, so I thought I'd just wrap up with like this, like 5 slides. It's really short. So first case here is batch effects. You will find them. If you haven't found them already, you will find lots of these. They happen. TCGA, all the big studies are full of them. Okay. What is a batch effect? It's some sort of systematic bias in the data. So it's not random. It's not stochastic. We can deal with that by increasing the sample size. These are things that are systematic biases. So some RNA extraction method, the technician that did it, the assay date, the date is a really big one. The machine it was run on. Okay? These are intrinsic properties of that data that we can't do anything about, and basically a whole load of samples have that property. Okay? Only experimental design can overcome these. So this is an example here. This is actually an ovarian cancer data from Mock et al. This is actually from Mike Beer, who we actually collaborate really closely, and I actually have analyzed this data in depth. So that's the reason why this is here. And this here is the date the assays were done. What do you recognize? But they don't all cluster by date, so it's not a clear pattern. There's just a couple of dates which are a little bit friendly with each other. Okay? But that will affect looking at variance. Now this is a much bigger problem if this group here were all a particular sample, these were the ones that were all treated with my drug, and these ones here were treated with the different doses of the drug. And that's a confounding covariate that we're going to get on to next. Rafa is going to be here next week, and he's going to talk much more about batch effects. And he will also, I think, present Combat, which is actually from Chingley and Evan Johnson, who's on this here, and surrogate variable analysis. Confounding covariates. This one is, tough one. This is down to poor experimental design. And this was a really exciting paper published by Landmain et al. In Cancer Research. And I saw this and I got so excited. I work in cancer and ovarian research, as I might have mentioned. And one of the things they had was they said that they had a signature of lung metastasis. Now, you know, to have a signature of a metastasis to a specific site from the primary is really like a holy grail in cancer. If we can say, when you present at diagnosis, and we take a sample of your biopsy and we say, you know, your cancer, you're going to have a biopsy, you're going to have mets to the brain, you're going to have mets to the bone, you're going to have mets to the lung. That would be really, really cool if we could predict that. That'd be just amazing. And so when I saw this, I thought, wow, this is really cool. They have 6 genes and they predict mets to the lung. Wow, 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 wow. And I looked at one of the genes and I went, I know this gene. this is a predictor of molecular subtype in breast cancer. I've done this. I've actually done a whole load of TMAs on this gene. I know it well. And I looked at their data and that was do. Because in breast cancer, you've got different molecular subtypes. So you've got the hormone-positive breast cancer, which tends to occur in older women, postmenopausally. Okay, it's associated with BMI, it's associated with different risk factors. You've got hormone-negative breast cancer, basal-like breast cancer. That tends to occur in younger women. It's non-responsive to hormone therapy. Very, very different case. Within the hormone-positive, then you have like low-grade and high-grade cancer, and then you've got a group which have RB2 amplified. Very, very different cancers. We treat them with different drugs. Just happens that they also have different predispositions to different sites. So if we look at lung metastasis, we get both the basal subtype and the high-grade hormone positives tend to go to lung. Okay? If we're looking at bone, we tend to get the hormone positive cancers tending to go to bone. They have different preference for different sites. In their design, they only looked at the basal subtype to lung. They didn't look at any other subtype, which means it's completely confounded. They're predicting the molecular subtype of the cancer, not necessarily mets. So we did a lot of analysis on this. This here is molecular subtype. This is their metastatic site. All of the tumors that they looked at that go to lung were basal subtype. We have tons of predictors that will tell the molecular subtype apart. This is a very, very easy problem in breast cancer. And these were their genes, and I went, no, we've just got a subtype. We did a whole load of statistics on it. It was completely confounded. We published it as a response to their article in Cancer Research. Because I was just like so gutted. But I wasn't impressed. That was published 2009. This is a really old slide, I'm sorry. That's a confounding covariate. It's something to watch out for when you're doing your experimental design, and sometimes it's difficult to do. Third case, and this is actually a paper that we're pulling together at the moment. This is an ovarian cancer. Ovarian tumors tend to be really large. Women tend to present with them with actual bowel problems in general. They tend to be asymptomatic and tend to get very, very big, and once they're like then encroaching on the other organs, people will say they're kind of feeling bloated and they're just kind of got some stomach upset. And at that stage, the tumors got very big, and what you tend to get, a lot of stroma cells or other cells in the tumor as well as the tumor itself. And because of this, when we actually look at ovarian cancer, we tend to have a mixture of the epithelial cells and the stromal cells. And this is a very large Australian consortium, a fantastic group, have actually published 6 subtypes of ovarian cancer, and also this has been verified by the Broad. We said, do you know what, because of all this stromal tissue in ovarian cancer, we should maybe go back here, just double check this. So this here is tumours of the same molecular subtype. We hypothesize if there's an admixture of different cell types, you may falsely discover subtypes. They're just basically associated with the fact that you have a mixture versus maybe something that's purer. Equally, maybe if you had a case here of where you actually have very different subtypes, but then if you actually sample those differently such that you have different amount of peripheral cells, additional cells, you may just increase this variance or the noise in your signal, and you may actually miss the discovery of important subtypes. So we've actually looked at this in a lot of detail, but this is from this analysis of the Australian cohort, and we looked at genes that were associated with stromal or tumour gene expression in microdissected data, and then we looked at their clustering. I don't like to say this because I think maybe there may be biology here, but one of the things that we are worried about is the fact that if you take genes that separate stroma and tumour, you actually find their clusters. This here is the 4 molecular subtypes of ovarian cancer. This here is C1 tumours, C2 tumours, C5 tumours, and C4 tumours. And these actually fall out by simply a gene signature which separates tumour and stroma genes. Now if we actually look at some microdissected data, and these were tumours microdissected from C1 tumours, see one of the black ones here, so they took a couple of these C1 tumours and microdissected them. If you look where the stromal component falls, it falls within the C1 component. If you look where the tumour component falls, it falls within the C4 component. So definitely stromal gene expression is driving a lot of the differences between these. We've done a lot more work on this, and this seems to be a systematic problem across ovarian cancer in that there's a lot of published gene signatures, including some with my name on it, and some with our collaborators. Actually, I think I'm on 2 of these papers. So this is not, I'm on this paper and I'm on this paper. No, I'm not. I'm on the second paper they did. I'm not that one. Where we've actually found that a lot of gene signatures that are coming out in ovarian cancer seem to be strongly associated with stroma gene expression. If you correct or adjust for the amount of stroma that's actually in the tissue, we begin to lose the prognostic significance of a lot of these signatures. And I think this is a problem not just in ovarian cancer, but in a lot of other cancers as well, where we're actually looking at mixtures of cells. So those are 3 different sources of variance and problems that you may identify in your exploratory analysis. The batch effects, confounding covariates, and also sort of biology and thinking about the biology and how that might be interpreted in the data. Okay. I will take a break. I'm going to do a little pop quiz on experimental design when we come back. It's only a 2-minute thing. Okay.